Thank you for joining us this morning. And today we have Dr. Russ Carmical, Director of Operations in Virology and Microbiology Department at Baylor College of Medicine. Dr. Carmical will be teaching us how to automate methods of 16S rRNA libraries, library preparations in next-generation sequencing. Thank you, Russ, for being here with us today, and I will turn it over to you.
Oh, very well. Thank you very much. I just want to give a special thanks to you folks that are tuning in for the second time and apologize for the technical difficulties we experienced at our last go-through. As Colby said, my name is Russell Carmical. I'm coming to you from the Center for Metagenomics and Microbiome Research at Baylor College of Medicine, which is located in the heart of the Texas Medical Center. I'm going to talk to you about our automated methods for both 16S and WGS library preparation. Before I dive into into the pipelines, I'd like to tell you a little bit about our center. Our mission is to understand how the microbiome impacts health and disease, and to translate this understanding for better therapeutics and diagnostics, and to serve as a hub for these activities internationally. Currently, we have over 300 collaborative projects with 185 collaborators at 74 institutions across the globe. So we've seen an exponential growth in the number of samples processed over the years, with just over 1,000 samples being processed at the year of inception, and nearly 70,000 samples last year. So I'd like to define a few terms for those folks that aren't familiar with this area of research, and that is that the microbiome is the entire collection of bacteria, viruses, and fungi that naturally occupy various niches on or in a given environment. And metagenomics is the use of modern technology, in our case, next-generation sequencing, to study communities of microbes directly in their natural environments without the need for isolation or cultivation of the individual organisms, which really has catapulted this area of research. So one of the most widely utilized tools in this area of research is the 16S ribosomal RNA gene sequencing. And the reason that we choose the 16S gene is that it's highly conserved. It's nearly 1,500 base pairs in length, but most importantly, it's the 9 variable regions that are contained in that sequence. So these act as a variable fingerprint, if you will, so we can identify the individual members in this mixed population. And so that information allows us to classify down to the genus level. So one of the things that we utilize is molecular barcodes, and that allows us to maximize the sequencing potential on the MiSeq or HiSeq platforms. And so for those of you that aren't familiar with molecular barcodes, the unique sequence that we incorporate in our PCR flanking regions of our PCR primer, and allows us to multiplex or to pool hundreds of samples in one sequencing run. So this type of sequencing is amplicon-based, so it's a typical PCR reaction, and where we focus is variable region number 4. You can see our amplicon size is around 290 base pairs in length. So what do we gain from this assay? First, we can tell who's there, so what members of the community, and then in what relative abundance are they there, and then how samples relate to one another in a PCOA plot. So now what I'd like to do is describe our pipeline. The samples are received in our nucleic acid extraction laboratory, and then once the nucleic acids are extracted, they're presented to our pre-PCR laboratory, and then the plate is sealed at that point, and then passed on to our post-PCR laboratory, where amplification and cleanup occurs, and then quantification of those amplicons, and then normalization prior to being passed on to sequencing. And you'll notice on the right-hand side noted are room numbers, and this is just to indicate that these major steps in our pipeline take place in individual laboratories, and that's to minimize the risk of contamination of primary samples with an amplicon. And I should mention that we operate in a regulated laboratory with CLIA CAP accreditation, and so we follow a unidirectional sample path so that samples are never going in the reverse direction to minimize that risk of contamination. So here's I'll take you on a kind of a virtual tour of the laboratories, the first of which is the Nucleic Acid Extraction Laboratory. And in this we have 3 Hamilton starlets that carry out our extractions. These starlets are 96-well fixed head platforms, so we'll do a plate at a time on each instrument. You'll notice that this starlet is outfitted with a UV light and a HEPA filter so that we can sanitize the instrument after we've run the extractions. This was available at the time of purchase for this particular unit, and we're having the others retrofitted. And that is a mandate for regulatory concerns. And then moving on to our pre-PCR lab, we have full-size stars. Unlike the starlets, these have 8 independent pipetting channels. A closer look at the deck layout, and so you'll notice that there's a number of tip carriers, tube carriers, our plate carriers, and then all the heaters and shakers that we need to carry out the library setup. And then you'll notice in the back we have a magnetic stand for bead cleanup, and then a barcode reader so that we can QC where we place labware on the instrument. So then moving on to our PCR laboratory, we have a bank of thermocyclers. And you'll notice in the right-hand corner, each of these is identified so that we can keep up with PMs and calibrations and monitor that for regulatory purposes. So once the amplification occurs, we pass it on to the post-PCR star, which if you notice that it has a very similar or identical deck layout to the pre-PCR, although it performs a very different task. And the reason that we did that is we need redundancy in our pipeline. And so if one of these instruments were to go down, we could sanitize with the UV light, and then it could perform the other one's function. So that kind of added the redundancy without purchasing 4 of these units, which takes up both space and funds. So in addition to the bead cleanup on this instrument, we carry out the pico green, so to quantify our amplicons. Once those are set up, they're taken off deck and read on a plate reader. That information is then fed back to the robot, which calculates how much of each of the amplicons should be added to the pool. So we want to normalize so we have a good even distribution of signal among the various members of that particular pool. And so you could imagine that's quite laborious with 400 different samples going into an individual pool. So then quite often when I give this tour of the laboratory, it's a little bit anticlimactic when we look at all of these elaborate liquid handling robots and we get to the little box that actually does the sequencing. But for those of you that have ever carried out any Maxim Gilbert sequencing and looking at the audio radiograph, you can appreciate the power of this box. So with the Illumina V2 kit, which we utilize, we can expect about 8.5 gigabases, or 8.5 times 10 to the 9th base pairs read. And then with the V3 kit, which is a 2 by 300, we can expect about 15 gigabases. So it's quite powerful. So then the obvious gain for all this automation would be throughput, right? And so what I've tried to depict here is how many samples we could process in a given day with one technician in our particular setup. And so on the left-hand side, you see the automated throughput. And rather than look at each of these steps individually, we just look at the lowest or the rate-limiting step. And so on the automated side, you'll see that the sequencer itself with 400 samples being pulled in a given sequencing run is the rate-limiting step. And then on the manual side, extraction is the most time-consuming with just 192 samples being processed in a given day. I would say more importantly than increase in throughput and productivity, because the technician now is able to go off and perform other tasks once they hit go on the robot. But the greater advantage, especially in terms of regulatory concerns, is the decrease in variability, potential human error, and contamination. So now I'd like to look at how we would go about validating this method. So I'll go through our experimental design. So we need a gold standard or baseline, and we chose samples that were sequenced back in 2013 through our manual preparation to be our gold standard. And so then we would then compare that to the automated method which we developed this year. So with any good experimental design, you want to look at what's held constant. In this case, it would be the DNAs, which were kept at -80 for the past 3 years. And then look at potential variability. And in this case, that far outweighs what was constant. So technicians change, PCR primers, kits. So it has the makings of a disaster. Any first-year graduate student would pick up on that. So we thought a little better about it. And we sent those same samples through our manual prep at the time that we did the automated prep. So with any good experimental design, we included negative controls, and we dispersed those throughout the plate so we can look for any potential cross-contamination in those wells. So when we look at the data, we should see no signal in those wells. And then, of course, we included a positive control. In our case, we used Tularensis francicilla, and we put that juxtaposed to a negative control so we can look. That's going to give us our hottest signal, so we'll look and see if there's any of that signal in our negative control. So we looked at, once we carried out the study, we looked at read counts, and I just chose some of the top samples in that pool. And on the left-hand side, you'll see that the negative controls contributed no signal, which is what we would expect and hope for. And then on the far right-hand side of the graph, you'll see our positive control, which gave us most of the signal. And then across these samples here, we get a very even distribution of signal, so that tells us that we did a good job at normalizing prior to pooling. And then the data output that we would look at would be observed taxonomical units. And so if we compare the manual back in 2013 to our automated this year, and then also the manual, you see that there's very little difference in the number of observed OTUs. Then plotted this against itself, so manual plotted against the automated, and we see an R-squared value of 0.86, which is pretty good considering all the variables that are involved in this type of study. And then we wanted to look at variance in the manual method itself, so we compared 2013 to 2016, and we came back with an R-squared value of 0.79. The idea here is to try to minimize the noise that is contributed by all the variables that we saw earlier and really get at the question, which is there any difference between the automated and the manual? Is there any bias that we should know about? So the next way that we look at the data is we look at relative abundance, and so I chose just top 18 taxa out of this analysis, and again compared manual to automated, or the two manual methods to the automated. And we saw very little difference in the relative abundance between these methods. And then the other way we look at this data is to plot it on a TSNE plot. And so what this does is reduces the data down to a single data point and then plots it. And so you'll notice that technical replicates plotted very closely to one another. I circled them here so you can kind of identify those technical replicates in the three experiments. We do see some outliers. You'll see the line is drawn between the technical replicates. In conclusion of that study, we saw that there is really no difference in observed OTUs, no statistical difference in the relative abundance of the taxa, and that the technical replicates clustered very tightly to one another. So again, since we're working in a regulated environment, we would want to produce a validation report before we could ever offer this to any collaborators that would be running clinical trials or any regulated study. And there's a lot of verbiage on this page, but I'll break it down for you. So out of the 393 original samples, we were able to sequence 384. You'll see that it's manual versus automated. So the first manual method in 2013 to automated in 2016. And so we found 380 mapped reads as compared to the original 393. And then if you look at the average number of reads, they're quite similar, and just over 25,000 average reads. So in this type of validation report, you want to measure a number of things. One would be accuracy, and so the question is, how do we define accuracy in a regulated study? And that would be the agreement between observed and expected, or map genera for our positive control. And so we found that 100%, we had 100% accuracy across the 4 plates for each study, which is no surprise if you remember the slide that showed the signal from our positive control. And then we want to look at precision, which is reproducibility. So we look at intra-run and inter-run variants. And that would be the concordance of OTUs, either with mock communities or generous donor samples, or any gold standard that you would choose. And we found that 380 out of the 384 replicates resulted in identical OTUs with a precision of 98.96%. And then the other thing that you need to look at is analytical sensitivity, which is the limit of detection. And so we kind of empirically derived this to be 1% abundance. So any sample contributing less than 1% would be filtered out. So then shifting gears to the NextEra XT validation study, we had a similar design, although we chose samples that were more recently sequenced. So we're again looking at manual versus automated. In this study design, we ran the automated study 4 times. So on the left-hand side of this table, you'll see the manual. We chose 4 previously sequenced, heavily sequenced samples from the HMP mock community. These would be individual isolates. And then we chose 6 isolates from our stocks. And then we ran a blank. And so once that checked out, we repeated those same samples in our automated method. And so if you look down and you say, well, 11 samples, that's not very high throughput, and that is true. We figured baby steps first. So we ramped that up a bit, adding 16 samples from our stocks. But more importantly, for a validation study, we added blinded samples, which we obtained from Rick Piles and Aaron Miller down at UTMB. And then we sequenced those and then obtained the sequence key from them after the study was run. And the other thing that we did was we added a mock community so we can get closer to a true sample source. And for those of you who aren't familiar with the mock community, we take individual isolates and then mix those in some ratio, a known ratio, so we should get that answer back once we sequence. And again, we're still at 36 samples, which is not very high throughput, so we ramped that up to 96. And then we ran these samples in triplicate to gain that 96. So for those of you that are familiar with the NextEra XT kit, you'll recognize these slender tubes that you receive in the kit, which are kind of odd little tubes and you're not quite sure what to do with. Illumina provides a nice plate or a base to hold these, but it's not very automation friendly. So the first hurdle that we had to clear was to get some type of adapter to fit these tubes into our tube rack carrier. And so Hamilton was kind enough to manufacture these and send those out so that they not only hold the tube but also present the barcode. Because as I said earlier, each of these robots has a barcode reader on them, which will check to make sure that the labware is where you say it's supposed to be. So it's a kind of a QC checkpoint. So just a little bit of background for NextEra XT for those of you that aren't familiar. It utilizes a transposase, which cleaves the DNA on average about 300 base pairs. And it also transposes an adapter sequence to the ends of those fragments. So once that's completed, it goes through a reduced cycle PCR amplification, which has the sequencing primer that anneals to those adapter sequences that were added by the tagmentation reaction. And then on the flanking ends, we have the index sequence, and then the P5 and P7 adapter, which allows those strands to anneal to the flow cell on any Illumina sequencing platform so that they can be sequenced. So once we've carried out the PCR reaction and the library setup, we want to QC those samples prior to committing those to sequencing where the true cost of this is. And so we utilize the advanced analytical fragment analyzer. So these are our readouts from that instrument. And you'll notice that we have a nice broad peak around 300 base pairs. So I will tell you our first run through with some of the samples were diluted in TE, which inhibits the tagmentation reaction. And so the bulk of our fragments were around 1,000 kb with or 1 kb, with some down on the lower end less than 100. And so that would bias that sequencing to those small fragments. So you really want to QC in some fashion, either with an FA or a bioanalyzer, prior to committing those to the sequencing pool. I should say another step in the NextEra XT, which is unique to this kit, rather than the mechanical, the bead cleanup and then the mechanical normalization of the pools, NextEra XT provides a plate with beads in them, so you saturate the beads and then wash off any excess. So in effect, that normalizes your sequencing pool at that point when you elute off. So then we look at that data with Metaflan, which was developed in Curtis Huttenhauer's lab at Harvard. So if you look at the top, you'll see an output from one of our single isolates. In this case, it would be Staph aureus. And so we got 100% readback mapping to Staph aureus, which was quite pleasing. And then at the bottom, you see a readout from one of our mock communities. And then again, that gave us back the answer that we expected with the appropriate ratios of those bacteria that were added into that mock community. So this is that data plotted out in a bar graph. On the left-hand side, one of the mock communities that we sequenced was the Zymo biopool. So obviously we have a known ratio and known members of that mock community to compare against. And so lo and behold, the data came back and gave us those individual members with appropriate ratios. And then also, one of the things we look at is the intra-run concordance. And so you'll notice that replicate 1 or technical replicate 1, 2, and 3 show very similar relative abundance of each of these bacteria. And then on the right-hand side, we see the mock community that we assembled, which was a staggered mock community. Staggered meaning that we added those in various ratios, whereas an even mock community would be added in equal ratios. And so, again, it gave us the answer that we expected, and then also the technical replicates are quite similar. So in conclusion of those validation studies, all known single-strain isolates returned the expected results, and then the unknown strains confirmed via inter-lab results. So we conferred with Rick Files' group down at UTMB for our unknown samples, and in fact, we came back with the expected results. And then mock communities returned expected results as well. So with that, let's look at our future directions. We'll continue to develop automated methods with Hamilton and Illumina. They've been great partners in these validation studies and provided us the reagents and the hardware that we needed and dedicated technicians to developing the methods for the robots. We're currently working with Genome and their Dragon Bio-IT processor. So we have developed a software package to analyze metagenomic studies, and they've developed a piece of hardware which is specifically designed to process next-generation sequencing data. And then also we're heavily involved with the regulatory agencies in developing mock communities and standards along with proficiency testing so that a core set of samples could be sent out to laboratories across the globe and sequenced and then compared back to a database so we can standardize this and hopefully move these studies into the clinic at some point. And then lastly, commercialization. So we have a commercial arm, Diversigen. So any samples that would come from a clinical trial, say from Big Pharma, would be funneled through that organization.
Thank you, Russ. So we have quite a few questions on the line, and I will start asking them. One is from Sharon Beckman, and she's asking you, how does the selection of variable regions can be to be sequenced affect the species identification? So, and to continue with another question was, why V4, why not V3 or V3, V4 together?
Yeah, so it shouldn't change the identification of the species. We focused in on V4, but we do run V1 and V3 as well, and even V5. So we've validated those studies comparing V4, which is our gold standard, to them, and we get similar results. It just so happens that our pipeline was developed around V4, so that's kind of why we stick with that.
Okay. Another question that I like, or I would have asked myself, is can you comment on your use of extraction controls to compensate for environmental kit contamination?
Great question. Yeah, so there's a fair amount of variability that comes in with extraction, but even prior to that, sample handling is a big issue. So the mock, I didn't go into the development of the mock communities, but the way that we develop those is we'll have a whole cell mock community, so we take stalks and grow those up and mix those in a particular portion. And then we carry out the extractions on those individual organisms, QC that DNA and mix up a DNA mock community. And so we'll run both of those through our pipeline and compare the data. And then as far as contaminations is concerned, we run the blank control with every run to look and see if there's any signal in those samples as well.
And as a follow-up question, is there a particular kit that you utilize to do this extraction?
We do. We use the Mobile kits,
okay.
and we use kind of a wide range of those.
Okay. So one question that was sent was, were the manual 2016 and the automated 2016 sequenced by single MiSeq run? And I had a similar question, actually, that was saying, how many samples do you multiplex onto a MiSeq run? That might actually answer it.
Oh, yeah, good question. They were, so, well, if the question, I should say, the automated method was on one MiSeq run, and then the manual was on a separate. So we pull, on average, 400 samples in a given pool, and so each of those contain 4 plates, or 96 whole plates that were pulled. The exception is low biomass samples. We tend to pull less, and then we also increase the amount of phi-x to give the variability that we need.
Okay. So another question similar to this one is, have you used multiplex negative controls in your automated pipeline to account for sample cross-contamination?
Multiplex negative, can you have them define multiplex negative? We don't. I'm not sure what that is.
So actually, it's multiple negative controls. So a few blanks, yeah.
We do. So on that plate, let's see if I can back up. Where is it? Yeah. So we add negative controls throughout the plate to look for potential crossover in those samples. And then again, one positive control that's juxtaposed to a negative, because the idea there is that's going to be the greatest signal contribution for the positive control. So if there were to be carryover, we should see it there. And also, we don't see tularensis in our samples, so if it shows up, we know we have an issue.
Okay. Now let's go to the bioinformatics. You were talking about the OTUs, and I was wondering which software do you use to actually get into your OTUs, and is your pipeline automated?
It is. So we have a team of bioinformaticians that developed something called Sage, which is available to people to use as freeware. Google, I don't have the link, but if you just Google Sage and Baylor College of Medicine, it should come up, either that or email me, and I can get that to you.
Okay.
I will tell you, I'm not a bioinformatician, so if we want heavy into analysis, I'll pull one of those guys in.
Okay, I'll just have one more bioinformatic question, is how about the beta and alpha diversity between automated and manual of the 2016?
So we looked at alpha diversity, which was plotted here, and see very little difference in the two, in the automated versus the manual. There's a few outliers, so we were quite pleased. As I said before, there's ton of variables in this study, different kits, different reagents, technicians, and so it's not surprising that we do see some variability. But we didn't use beta diversity as metrics for this validation study. And so this is how we see validation. This doesn't mean it's either the right answer or the only answer, so I'm always open to other suggestions. I gave a talk at NIST about a month ago, and so we got lots of good feedback there. We're working with our bioinformaticians to kind of tease out what is the best way to evaluate the question that we want to ask, and that is, do we get the same answer each time that we run this? Because regulatory agencies want to see that if 5 years from now, if I come back, can we run the same samples and get the same answer? And so you need a version control on your software, which we do, because it does change. I mean, it's a pretty dynamic field and things are changing all the time. But we're very careful to timestamp those so that if somebody came in and wanted to repeat the study, we could do that. That answers the question.
Yeah, it does. I have another very interesting question. Was there a particular reason why you chose the MiSeq DX instead of RUO MiSeq?
Good question. So the reason that we chose the DX, again, being in a CAP-regulated environment, is I needed the performance qualification, which is a very time-intensive document to generate for yourself, and so that came with the DX model, so that's why we chose it. But you could obviously you can run these studies on a regular MiSeq as well. In fact, we run it in RUO mode. We don't run it in the DX mode. The decision was purely around that document.
Yeah. So one quick question, why did you select Nextera XT versus Nextera as a library prep method?
Yeah, good question. So the reason, obviously, Nextera XT is developed for small genomes, and so it was appropriate for these samples that we're looking at, but also it was a need that Illumina had to develop an automated method for this kit, and so that was the reason behind it.
Okay. And then how much PHI-X did you use to get for your samples, usually on your MiSeq run?
Yeah, that's a great question. We typically run about 4%, which is much lower than what Illumina recommends. In fact, when we have any issues and we call tech support, they question us as to why we're not. But it eats up a fair amount of sequencing real estate, and so we keep that to a minimum. Now, with low biomass samples, we will increase that to up to around 15%.
Okay. Well, thank you so much, Russ.
Oh, my pleasure. Thanks for having me.